Embark on a journey of inspiration and discovery with the Purdue Lecture Hall series, proudly presented by the Purdue Institute of Inflammation, Immunology, and Infectious Disease. Join us as we delve into the remarkable odysseys of these aspiring scientists, each crafting their own narrative in the world of science and groundbreaking research. Take a glimpse into their diverse cultural backgrounds and the journeys that brought them to Purdue University. Thank you for watching the Purdue Lecture Hall series. Remember to hit the like button and subscribe to our channel. Welcome, everybody. My name is Tommy Soares. I'm the Director of Scientific Strategy and Relations for the Purdue Institute of Inflammation, Immunology, and Infectious Disease. And today on our Purdue Lecture Hall series, we have the pleasure of welcoming Eylem Kulkoyluoglu Cotul, earned her bachelor's degree in molecular biology and genetics and a double major in chemical engineering from Istanbul Technical University in 2013. And that was followed by a master's degree from Koc University. In 2015, Eylem moved to the US for her PhD at the University of Illinois at Urbana-Champaign, completing it in 2019 with research on metabolic plasticity in hormone-positive breast cancers. After receiving her PhD, she worked as a postdoctoral researcher under Dr. Michael Wendt here at Purdue University in the Department of Medicinal Chemistry and Molecular Pharmacology. And she's been focused on metabolic changes in obesity-related metastatic breast cancers. Currently, she moved from Purdue and is now a research scientist at Immunome Incorporated, contributing to immunology research and innovative therapeutic developments. Welcome, Eylem. Thank you so much for taking time to talk to us. I am so excited to hear this story because I know that your journey has taken you far, but I think our audience is so interested to understand this progression and now you moving from the postdoc to very recently working at Immunome, a company close by. And we're so excited to have you on the show. Thank you again for coming on.
Thank you so much for your invite, Dr. Soares. It's my pleasure to be here today. I think we are ready to start.
Yeah, let's do it.
About my journey.
Absolutely. So go ahead, share your screen. As typical, I'm going to turn myself off here, let you take the stage and the spotlight, and we are all ears. So if you want to share your screen, I can give you confirmation of what it looks like. Perfect.
Can you see my screen now?
I can see your screen.
Perfect.
Awesome. Thank you.
My pleasure. Hello everyone. My name is Eylem Kulkoyluoglu Cotul. Today I will be talking about my education journey and also at the same time my career journey, let's say. So this is like, I wanted to include this slide as a brief outline about, who I am, my PhD, my postdoc work, a little bit about my previous work before PhD, and my current industry experience at Immunome. So, like I said, my name is Eylem Kulkoyluoglu Cotul. I am originally from Istanbul, Turkey. And as a brief introduction about where I am, so Turkey is a country, and it is located as a bridge between Asia and Europe. So this is the location and this is our flag. So basically, I am here almost 5,500 miles away from my home country. But yeah, I wanted to bring insights about where I am from first, and I wanted to start with start introduction with this information. And if we wanna walk through, what kind of touristic attractions we have in my home country. So the most important touristic attraction is obviously Istanbul. Istanbul is located on the north west of the Turkey, and it is located here. And this is where I am originally from. And actually, I took these touristic attractions and all these places from Lonely Planet website because I don't want to be biased. So the second most important touristic attraction in Turkey is Cappadocia, definitely. This place is located somewhere around, between Aksaray, Niğde, from that area, and it is a beautiful landscape. Like, if any of our audiences have a chance to visit Cappadocia, I think it will be a very nice experience for everyone. And other important touristic attraction is definitely like Mediterranean coast and Aegean Sea coast. So we have beautiful Mediterranean Sea coasts and Aegean Sea coasts. Again, if any of you have a chance to visit Turkey, definitely these places are need to be seen. And Pamukkale travertines, I wanted to include this place too, because this is I should admit that I am a little bit biased about it because this is where my father is originally from. So I think this is a really important landscape. And it's a really interesting place. So it is naturally warm water, and actually these places are completely natural. This landscape is completely natural. I think this place needs to be seen too. Definitely our northeast coast, on the Black Sea side, it is also worth to see. There are lots of things to worth seeing there. So we have beautiful mountains, beautiful landscapes there too. It's a little bit rainy, but if you like nature, I think this will be a really nice place to visit too. And the last place I wanted to introduce is our southeastern Anatolia. It has beautiful um, um, history, beautiful landscapes. Again, if you are interested in history specifically, people should go and visit our southeastern part of Turkey as well. Again, I also wanted to include my hometown. Again, I am a little bit biased on that side because Istanbul is where I am from and where I spent my childhood and college years. That's why it has a really, really big importance in my life. Again, when someone says Istanbul, I think the most important places will be like Grand Bazaar, and Spice Bazaar at the same time. Our Bosphorus Bridge and Bosphorus region, Süleymaniye Mosque. This is called like we have so many mosques and beautiful landscape. Hagia Sophia, this is also very important for I think everyone. Galata Tower and Istiklal Street. These are a few important places to be visited in Istanbul. That's why I wanted to include them in a special slide, I would say. And like I said, Istanbul is where I am from, but my career journey started with a red microscope. I think it was my fifth or sixth birthday. I don't remember it very well, but my uncle Ergun Nalbantoğlu gave me a present. Gave me this microscope as a birthday present, and I think that changed my life, because I was spending at least 2 hours without any break with this microscope. I was just collecting um, um, samples from flowers, from different plants, etc. And I was just, too curious to look at them under microscope. That's why I'm still keeping this microscope. It is in my parents' house, and I can say that this microscope and my uncle indirectly changed my life, and he gave a start to my career, I would say. And at the same time, my other uncle from my father's side, Okan Kulkoyluoglu, he's also a biologist. He's an environmental biologist, and he was pursuing his PhD when I was like 6, 7 years old. And that's why I was always like, he was my role model to be a scientist. And definitely my parents, they put so much effort on my education, and they put so much effort on my development as a scientist. That's why I wanted to include them here too. And lastly, my husband, Uğur Çatul, now he's my husband, but before he was my college friend. So he witnessed all steps in my career. That's why these people constructed. I can say that these people encouraged me to be a scientist, and they started my career journey. Some of them started my career journey, and some of them supported me at every step. That's why I'm always so grateful for their efforts on my career and on myself. Like Dr. Soares mentioned in his introduction, I took my bachelor degrees from Istanbul Technical University. I also wanted to include a few photos from my second home, I would say, because I always consider Istanbul Technical University as my second home. This is where I spend most of my time, and I am so grateful for the education that I took from this university, because this it was exceptional. It was our education curriculum was accredited with international institutions, and we were, I would say, developed as a real scientist in that school. And also, it was really important for me to be an Istanbul Technical University alumni, because they provided me a chance to do an internship in Munich Technical University for 1 year. So Istanbul Technical University and Munich Technical University were kind of sister schools, and they had an agreement between each other. And by taking this advantage, I was able to spend 1 year at Munich Technical University School of Life Sciences in Freising. And that was the most important and valuable step in my career, I would say. So here at TUM, Munich Technical University, I think it was my first real experience to be in the lab by myself, running experiments, learning steps of different experiments, learning different steps of molecular biology techniques. That's why it plays a really, really important role, critical role in my career. I think this is where I learned to be a real wet lab scientist, I would say. And I wanted to include Dr. Kai Schneitz here because he was so brave. He was, maybe, he doesn't remember me anymore, but he gave me a chance to spend 6 months and learn all the molecular biology, learn and execute all the molecular biology techniques as a third-year molecular biology and genetics student. As an undergrad, it was an invaluable experience. And also, I am so grateful to all the members in his team, because they provided me like a lot of experience. They trained me so well. They were never shy to share important details about molecular biology technique executions, etc. That's why I consider TUM um, uh, like very, very important in my career. That's why I'm so grateful to Dr. Schneiss for accepting me to his lab. And I also took, so after this 1 year, I was close to my graduation. And by the time I was trying to finish my chemical engineering minor, and so I think it was in 2013, I started my master's degree at Koc University. Koc University is one of the most successful private universities in Turkey. It is a very prestigious university, and I am also very grateful to be a Koc University alumni. And also, specifically, I am so grateful to my PI, İbrahim Halil Kavaklı, Dr. İbrahim Halil Kavaklı. It was my first professional experience as a researcher, as a molecular biologist, and I spent 2 years in his lab. He is a great mentor. He is a great human being. He is one of the nicest people in my life, and we are still in touch with him. I learned too much from him. That's why I'm also very grateful. Like all his support for my career. So he is a great mentor. He put so much effort on my development as a researcher, but at the same time, he was helping me to achieve my goals, because I always wanted to pursue my PhD in the US, and develop my skills further here in this country. And he was always coaching me to make my dreams true. That's why I am so grateful to him. And also I am so grateful to my beautiful lab mates in Kavaklı lab, because they are like lifelong friends. I am so happy that I know each of them. They are like whenever I think about them, I get super emotional, because they are like They are like my sister's brothers, and we are still in touch. We are still talking and sharing some insights about our lives. That's why, you know, I am very grateful to Dr. Kulkoyluoglu for accepting me as a master's student in his lab. But at the same time, so this was important for to learn more about his research. But at the same time, I'm so grateful to him to uh, like enable me to know all these beautiful human beings. And our research was directly related to circadian rhythm at Kulkoyluoglu Lab. So we did a lot of circadian rhythm research, and my master thesis was directly related to um, related to a protein which has an importance in both circadian rhythm and metabolic regulation. And after that, in 2015, I moved to the US to pursue my PhD. I came to Dr. Zeynep Madak Erdoğan's lab, and this is where I started my PhD journey. So University of Illinois Urbana-Champaign, which is not super far from here. So it is only almost 1.5 hours. Again, I spent there like 4.5 years, and it was really, it was, you know, one of the most important research experience in my career. So this is where I developed my niche experiences and research interest. So this is where I started to work on breast cancer. So the oncology research was always my dream because my grandparents passed away, died from cancer. That's why I always wanted to be a cancer researcher, and this was the address that I went after my dreams, and I think I took the most important step to be a cancer researcher here. So this Zeynep Madak Erdoğan's lab was, so the main focus at Zeynep Madak Erdoğan's lab was women's health and metabolism. That's why I learned like almost everything about oncology research, and I also developed my knowledge in metabolism in this lab. And again, I am so grateful to Dr. Madakardan because she accepted me as a PhD student. And I should also highlight that I am her first PhD student. That's why it was like, as a researcher now, when I evaluate the situation, it is really risky to accept someone as your first PhD student to your lab. That's why she took this risk. I hope it turned out to be a good experience for her too. But it was an invaluable, it was a great experience for me. And also, again, I made a lot of lifelong friends here. Alicia, Ashley, Chen Ying, and my brother Ozan. They are always like, we were always like considering each other like sisters and brothers. We were not like, we were not only lab mates for each other. That's why, you know, this was a really important factor for a PhD student's life. If you have encouraging, if you have really nice lab mates, I think it makes a really big difference on a PhD student's life. That's why definitely research is important. Definitely your PI is important, but I can say that as a graduate student, please go, see, meet, and talk to your future lab mates. This is really, really important for a PhD student's life. And I actually wanted to go a little bit deeper about my PhD research. The title of my PhD project was The Role of Metabolic Rewiring in Endocrine Resistance. Like you see here, I was working on breast cancer specifically, and I was the subgroup of the breast cancer cohort I focused on was endocrine resistant breast cancers. That's why, and I was specifically interested in hormone positive breast cancers during my PhD research. That's why estrogen was playing a really, really important role on my research at every stages of my research. That's why first I wanted to start with talking about estrogen receptor. So this is a figure from one of my thesis chapters. So we all know that, more or less, we all know that estrogen receptor plays a really, really important role on breast cancer initiation, progression, initiation and progression. And these are the important, I don't want to go into detail about my research, but in general, I can say that estrogen is a major regulator of breast cancer cell phenotype. And it is important, and I can highlight this importance when we look at different subtypes of breast cancers. So basically, in clinic, the treatment of breast cancer starts with, most of the time, I would say, this is like in general, definitely there are, a few exceptions. So breast cancer treatment starts with surgery. Then if it is needed, it continues with radiation chemotherapy, endocrine therapy agents, or targeted therapy agents. So these, different therapy techniques or treatment techniques are clinicians make their decisions based on this molecular subtyping of breast cancer. So they take a biopsy from the actual tumor and they send it to pathologists to make this subgrouping. And there are basically 4 different subgroups, like I said, like in general, 4 main subtypes of breast cancer. So luminal A and luminal B subtypes are hormone responsive subtypes, and almost 2/3 of breast cancers are hormone positives, hormone positive or estrogen receptor positive, I would say, if I need to be more specific. Therefore, estrogen has a really, really big importance in my PhD research, because most of the breast cancer patients are estrogen receptor positive. And even today, tamoxifen, maybe you heard about this medicine. So tamoxifen is an important and gold standard targeted therapy technique for specifically, especially for premenopausal patients, and also other patients too. So it directly targets estrogen receptor in the cell, and estrogen receptor goes and binds to estrogen receptor elements in the nucleus, and it basically normally excess estrogen binds to estrogen receptors, and it promotes cell division, proliferation in cancerous tissue, but tamoxifen binds and inhibits, like blocks estrogen receptors, and it blocks cell proliferation eventually. And this is a really useful drug because it is orally active. So it is like you don't have to take this drug like intravenously. It is really easy to use, and it is even today, almost 50 years, 50, 60 years after its discovery, it is still a gold standard therapy technique. But unfortunately, when breast cancer patients treated with like surgery and followed with tamoxifen treatment, in the 5, 10, or 20 years after this treatment regimen, their breast cancer tumors can relapse, and most of the time this relapse occur because of tamoxifen resistance. So my core topic of PhD research for my PhD, for my dissertation, was to come up a way to overcome tamoxifen resistance in this resistant patient cohort. And there are many. I was specifically identifying different ways of tamoxifen resistance development. And you know, when you look at the literature, there are many ways that cancer cells, breast cancer cells, can develop tamoxifen resistance. It can be through metabolic alterations. It can be through overexpression of drug efflux transporters. It can be through changing phosphorylation patterns of important kinases. Again, I don't want to go into that detail, but in general, my take-home message from this slide was there are many different ways that breast cancer cells can develop a resistance mechanism against tamoxifen treatment. So that's why we were trying to come up with an alternative way to overcome this resistance. And when we were making our literature search, we came across with another drug called selinexor. And I was specifically testing the effect of these individual tamoxifen, individual selinexor, and combination of tamoxifen and selinexor effects on different breast cancer in vitro and in vivo models. So if we look at this slide, selinexor is directly targeting XPO1, which is an important protein, which is an important export protein in the cell. And this XPO1 is important because it exports protein in the nuclei to cytosol. And when we look at patient statistics, clinical data, all the patients who have higher XPO1 gene expression have lower recurrence-free survival rates. So that's why inhibition of XPO1 protein is really important for breast cancer patients in general. And in the first chapter of my thesis, we looked at we try to identify, understand how XPO1 overexpression is associated with tamoxifen resistance in breast cancer. To be able to do that, we generated our tamoxifen responsive and tamoxifen resistant cell lines, and we saw that our experiment results show that whenever we have XPO1 protein overexpression in the cell, this overexpression of XPO1 makes breast cancer cells proliferate, grow more aggressively compared to tamoxifen responsive control group. And in the second part, I looked at, I changed my gear a little bit to metabolism, and I looked at what is the reason behind tamoxifen resistance development, and what is the reason behind selinexor only treatments inefficient results. But when we combined tamoxifen with selinexor in these tamoxifen resistant cell lines, we were able to decrease their metabolic rate. We were able to decrease metabolic rate of these breast cancer cell lines, and we were able to make these cells more autophagic, and by autophagy, these cells, we were able to overcome tamoxifen resistant in the resistant cohort. And in the third chapter, in the third part of my PhD thesis, we focused on more how this combinational treatment, tamoxifen and selinexor combinational treatment, affect on metastatic type of breast cancers. And excuse me, and we found out that this combination treatment is really effective to block metastatic progression in and in our breast cancer models. And overall, all the work we did was directly translational into clinic. And it has a really important role because whenever all the experiments, whenever I was like trying to get a result, I was always really excited about my PhD research, and I know that I was doing this to save some lives. That's why I was so motivated. I was working like, and I wasn't complaining at all. I was working and I wasn't complaining at all because I was trying to make an impact on people's lives, on patients' lives. And these are basically, you know, super detailed, but what I want to say here, yes, all these models, all these, you know, human models are a kind of model here, but they were not just numbers, not just models, not just, you know, sample numbers, according to my understanding during my PhD research. I was always considering each one of these people, each one of these women, as an individual, as a mother, as a daughter, and as a wife. And I was always trying to go beyond my limits, my physical limits, to work harder and harder and to achieve something. And good work paid off. We were able to so this selinexor as a, you know, drug, it was able to get an FDA approval for some cancer types, and definitely, you know, I'm not saying my research is directly the reason behind this FDA approval. Definitely it is like I'm not the only person to make this, but if I was able to put even a little bit effort on this approval to change these people's lives, you know, that was really important. And also, I need to go back and thank to my PhD advisor, Dr. Medakardan, because she gave me that insight. Yes, we are working in the lab, we are working on cells and sometimes, mouse models, but we are making, also our basic main aim was to make a change on people's lives. So she gave me that insight. That's why I'm so grateful to her. And right after my PhD, I came to Purdue. Again, I didn't go so far. I came to Purdue University. I decided to pursue my postdoctoral training at Dr. Michael Wendt's lab. And Dr. Michael Wendt was also, he he he was a great mentor. He's a really, nice human being. Like I'm so lucky because all my PhD advisors, they are like, they were all exceptional. All my mentors are, during my career, they were super supportive. They were trying to give me main insights and details of our research, and they are trying to train me as a researcher. That's why I am so grateful to my master advisor, my PhD advisor, and also my postdoc advisor, Dr. Wendt. The research at Wendt Lab is directly related to metastatic breast cancer. So here, FGFR1 pathway, EGFR pathway, HER2 targeting therapies, and tumor dormancy were four main topics, four main research interests at Dr. Wendt's lab. And this is our lab. Again, I'm also very grateful to Dr. Wendt because I know, beautiful people. I was able to work with, beautiful, smart researchers at Purdue University. Like, and also she's not here. You can't see her here, but our previous postdoc, Amara Abdullah, she put so much effort on my training when I first started my postdoc journey at Purdue University. That's why, together with Dr. Wendt, I also wanted to include her in my talk. And here, all my lab mates, they're like, most of most of the individuals in this picture, they already left the lab and they continue their research careers successfully. And yeah, so they are like, it was a really, really good experience to be in this lab because I learned a lot about different mouse models, different techniques, and I developed my insights for grant writing, for lab management, all the specific duties of expected from a postdoc were checked when I was leaving this lab in 2013. That's why I am so grateful to Dr. Wendt. And I also wanted to share important our previous paper So it was just published like almost 15 days ago. So definitely as a postdoc, I work with different projects during this 4 years at Wendt Lab, but this was the most important project I work with in this lab. So this was my main project, and I'm so glad that um, it turned to be a publication, because this is an important record for a postdoc's career. So, okay, I'm sorry. So the title of the title of my main postdoc project was the role of FGFR1 signaling in obesity-driven pulmonary outgrowth in metastatic breast cancer. So as you can understand from the title, I was still working on breast cancer, but this time I focused more on metastatic insights and metastatic subtype, a subgroup of breast cancers. And in this paper, we try to understand the role of FGFR1 signaling in obesity-driven pulmonary outgrowths. So basically, there are three different parts in our research. So the first one was FGFR1 signaling. Second important point was obesity, and the third and main part was the metastatic breast cancer. And when we look at a brief introduction about this research, so definitely obesity should be the number one to start with, because we know that, and also there are like super solid um, um, literature, information in the literature, and this literature, and all the breast cancer literature show that obesity is obesity decreases the survival of breast cancer patients. So and it is not only for one subtype, it is important for all four different subtypes of breast cancer. That's why, so this was our first motivation point to start a research related to obesity. And the second important part for this paper is FGFR1 signaling, because again, when we look at the literature is very clear that fibroblast growth factor, FGFR1 signaling, is a critical pathway for cellular proliferation. And literature was also showing that obesity and excess energy activate FGFR1 in breast tumors through FGF1 produced by hypertrophic adipocytes. So obese people, they have more adipose tissue accumulation in their body, and this excess adipose tissue is the source of the ligands for FGFR1 receptors. So it is like working as a wish cycle. So if an obese cancer patient, so if an obese individual is diagnosed with breast cancer, it is more probable to see recurrence or metastases in these patients compared to their lean counterparts. So this was an established knowledge, information in the breast cancer field. So the objective in our study was, I also wanted to check my time. Okay. So the important insight was, definitely in the first part, we tried to develop different mouse models to mimic obesity profile in mouse. And also to be able to do that, we were injecting our we were injecting our mice with different types of breast cancer cells, and eventually we saw that uh, people or the mouse fed with more fat, we see more metastatic nodule formation in the lung of these mouse compared to their lean counterparts. So this was important, and as you can see, we see really big mass on samples fed with 60% diet, 60% fat including diet. And also, again, I wanted to skip details, but we had to develop some tumor models to study obesity in metastatic breast cancer, because so the hormonal status and environmental temperature were two important points that we played with to generate our novel tumor models. So, excuse me. So ovarectomy and thermoneutral housing allow diet-induced obesity in a specific type of tumor model in our research. And again, the third and one of the most important part in our research was also related to dormancy, because so dormancy means a person can have breast cancer, can be treated, and after after treatment, even after treatment, some tumor cells can left in his or her circulation for some time. So we know that circulating tumor cells are they have potential to form metastases. They have potential to form distant tumors and recurrence. So that's why and our research, in our research, we also wanted to include dormancy models, and we saw that obesity is a diet-induced obesity prevents pulmonary dormancy in our models. And now, after that, we wanted to go a little bit deeper, and we wanted to understand how FGFR1 signaling contributes to obesity-driven breast cancer. And we saw that diet-induced obesity increases serum levels of FGF2 in biopsy mice, which is important to relate FGFR1 signaling with metastatic breast cancer, obesity-driven metastatic breast cancer. And as you see here, we did like so many tests on mice in vivo and in vitro, and the overall conclusion of our research was FGFR1 signaling can prevent pulmonary tumor dormancy in this obesity, diet-induced obesity driven metastatic tumors. So there are two take-home messages. If I wanted to put everything together, there are two take-home messages from my main postdoc paper. First, obesity increases systemic concentrations of FGF2, which is an important ligand for FGFR1 signaling. And second was, so this FGF2 accumulation acts on FGFR1 expressing metastases to break dormancy and increase outgrowth in the pulmonary side, especially in the lungs. And this was, I tried to make a, summary of my postdoc paper too. And again, so I recently, like 5 months ago, I started to work for Immunome Incorporated, and this I started another journey here because still we are interested in oncology research, but not only for breast cancer, we are interested in many different cancers, and we are trying to come up with novel transformative targeted therapies. And these therapy options include immunotherapies, targeted effectors. We are trying to design radioligand therapies and antibody drug conjugates. And I am so grateful to my supervisors at Immunome, because they are always supporting me to gain more experience and develop my skills as a researcher. And finally, I wanted to include this. So I am a lifelong learner. And it was always so important for me to continue learning and continue being productive during my career. That's why I decided, so, while I started working for Immunome, I also decided to pursue another master's degree in biomedical engineering at Purdue. And I recently I had a chance to work with Dr. Luis Solorio. And right now I am taking different courses, learning deeper insights of biomedical engineering, and I am enjoying learning so many different things and extending my skills as a researcher. That's why I am also so grateful to Dr. Soloria for accepting me as a full-time worker, as a student, as a master's student to his lab. And finally, I wanted to thank you for your time, and I wanted to thank Dr. Source for giving me this opportunity to share, all the insights of my research.
Well, thank you so much, Eylem. That was great. And I tell you, from the way that you put it, I really need to book a trip to Istanbul and to Turkey, because it looks like it's so beautiful. And also, what a journey. I thank you so much for taking us through it, and with passion, with emotion. It's really wonderful to be able to go along some of that experience that you've had. And to also see you so determined to continue learning, and not only, working at a company, but then coming back and saying, no, I want to continue improving my skill set. So congratulations.
Thank you
I
so much.
think, all I can say is best wishes, and congratulations on all your success and your hard work. I should say, Dr. Eylem Kulkoyluoglu Cotul, thank you so much for providing us with your take today and your journey. It was awesome.
Thank you so much, Dr. Source. I appreciate your time as well, and I appreciate giving me this opportunity to share, and thank to everyone important during my career journey. Thank you so much. It was
Absolutely.
a
Well,
pleasure.
thank you, Eylem. I hope you have a great rest of your day.
You too. Thank you so much. Bye-bye.
Bye-bye. Thank you for watching the Purdue Lecture Hall series. Remember to hit the like button and subscribe to our channel.